Hello everyone, in this video lecture we will see a tool which is used to predict mainly the three-dimensional structure of mutated proteins, and it is known as PHYRE2 tool. PHYRE2 is a suite of tools available on the web to predict and analyze protein structure, function and mutations. It uses advanced remote homology detection methods to build 3D models, predict ligand binding sites, and analyze the effect of amino acid variants, for example, non-synonymous SNPs for a user's protein sequence. The link which we may use to access this tool is given here, or you may also search it in Google. When you will click on this link, it will open the home page of the tool, where you can see on the top left the name of the tool, and then you are able to see here some boxes or windows. Like one is provided for the email address, where you will provide the email address where you want to receive the results of this job, and then there is a job description, and then there is a big window, larger window, in order to paste the sequence of the amino acid. So I have given my email address and then job description, and also pasted the amino acid sequence of my protein, which is FADA protein from bacteria, while the rest of the some of the parameters like modeling mode and for profit, non-profit, you are not supposed to interfere with these parameters, as they will be used like as said by default. And then you will click on the file search, and this will give you the job status. This will show you the job status, like in at first it is performing the analysis, like finding the homologs with PSI-BLAST, which is the job which it is performing right now is given in orange color. And then after some time you can see the job has changed, which is now building hidden Markov model of sequence. And then the job is checking for transmembrane helices, then loop modeling. Now you will get the email, through the email, the link for your results. I can show you that here I got this result for my protein FADA, and let me show you. When I clicked on the link, the page opened is shown here. I cannot show this page you as a whole on the PPT, so I just separated it into parts, but I want to show you here as well. At first you will get the information regarding your query sequence, and you may also see here the job expiry, which can be renewed for 30 days. And you may also download the FASTA sequence of your job, and then you may download the whole results by clicking on this button. And at first you are able to see on the left side the picture of the three-dimensional structure of your protein, and this structure is predicted by the PHYRE2 tool, and its three dimensions along X, Y and Z are also shown here. You may download this protein sequence from this point. And then after 3D structure, in addition to 3D structure, it also predicts the secondary structure and the disorder of the protein amino acids, which is shown here. This is actually a better picture which can be presented in your manuscript for the 2D structure. Although I have told you about the SOPMA tool, which is also used for the secondary structure interpretation of the proteins, and that is much better as compared to PHYRE2 with reference to the 2D configuration, because here you can find only the two attributes of the secondary structure, that is alpha helix and beta strand, but the SOPMA tool can provide you with additional attributes of 2D structure. However, the picture given here is much better as compared to the SOPMA tool for presentation in your manuscript. So the choice is yours, and here you can see the part which is given in green color is showing the alpha helix, and the arrows in light blue color are showing the beta strand, and its percentage in the whole protein is also given. And then there is domain analysis, like the tool has predicted multiple domains in our proteins, and the aligned regions are also shown here. You may click on the aligned region, and it may give you the specific molecule with which it has aligned. Like here it is showing the alignment with fatty acid oxidation complex subunit alpha from E. coli. And then there is second domain. So you may also present these domains in your manuscript. And here the as we have as I mentioned that it is a homology modeling tool. So you can see the model, the aligned coverage of this of the predicted model, and the protein with which it has aligned, and the confidence as well as the percentage identity. Now at the end of this page you will be able to see the topology analysis. As the protein I have given to this tool for to analyze did not contain any transmembrane helix or the topology, so we didn't find any topology results here. But in case if your protein has the transmembrane helices, then you will be able to see it at the end of the page of your results, and it will be shown, the picture will be shown like this, in which the two yellow rectangles are showing the transmembrane helices, and the dark gray structure is the membrane, part of the membrane. Now I have just presented these results by splitting them into parts, as it was not feasible to show the whole results at once. And now how can you present the data? How can you present this data in your manuscript? You can perform the analysis first for your normal protein, and then one by one for your mutated sequences, and you can copy the 3D structure and then paste it in the form of figure. Like at first you will paste the sequence of your normal protein, and followed by the sequences of your mutated proteins, like from case 1, and then from case 2, and then case 3, and so on. And it will make it easier for the reader to see the whole 3D structures at once, and compare the structures between the mutated and the normal protein. And how will you represent your topology? For that I have represented some of the results from my published article, and you can give the heading effect of SNPs on transmembrane topology of mutated proteins. And here at first in the figure on the top left side I have shown the picture for the normal protein, which you can see that it contains two transmembrane helices, while on the top right and on the underside, these are the topologies for the cases 7, 9, and 10, and which you can see they are different from the normal protein, because in the top right in two of these there is only one transmembrane helix, while on the left side you can see there are three transmembrane helices. So it is making very easy for you to represent your data through comparison in the normal protein and the mutated proteins. And also you can see here the numbers which are mentioned in each yellow rectangle, and these are showing the number of amino acids which are making this particular transmembrane domain. Like in this in one case 207 and 222 is mentioned in the same domain, so it is showing that this domain is comprised of the amino acids ranging from 207 to 222. So this was all about the PHYRE2 tool. Thank you so much.